put the first slide, please? Yes, that's all. So, hello everyone, thank you for being here in this workshop number 7 at ICOM. I am César Marín and I am co-chairing this workshop with Nancy Collins Johson. Today we have a very varied and very mixed plethora of speakers, mixed not just in the topics, which will range from nuclei level selection to plant holobionts, but also mixed regarding career stages. We have 2 PhD students and 2 very accomplished professors like Nancy and Bever, and then me, which I don't know how I fit into that. And yeah.
So it's my great pleasure to introduce our first speaker this afternoon, and that's César Marín.
So, this talk is entitled Tripartite Framework for Defining Units of Selection in Mycorrhizal Symbiosis. You will see later on which tripartite I mean. It's not like a tripartite symbiosis, although we are speaking about that, but it's a tripartite concept. So first of all, many thanks to the people that have inspired this seminar, this presentation. Nancy mainly, but also 2 very accomplished philosophers of biology, Javier Suarez from Spain and Elizabeth Joy from Indiana University. I have been speaking with them a lot the last 2 years, and it's very challenging, it's very nice. Actually, last year I went to Oviedo to teach a class in mycorrhizas to philosophers, which was incredible. Well, my talk is divided in 4. First, very briefly, I don't think that I need to remind this audience a lot about this, but that mycorrhizal mutualism is context dependent, then multilevel selection theory, then units of selection in mycorrhizal symbiosis, and then instead of conclusions, I will have like open questions, really. So, mycorrhizal mutualism is context dependent. This morning, 2 of the talks mentioned this, that in the functioning of mycorrhizal symbiosis, we have a continuum between mutualism to parasitism, and this has been proved a lot. There are very nice meta-analyses showing this concept, like this one by Johson and co-authors from some years ago. What this study has shown is that mutualism is enhanced under limited soils regarding nutrients. Then more recent research by Stephanie Luce from Agroscope, they show that mutualism is also enhanced under pathogen stress. This was a very nice study from last year. They inoculated 54 corn crops in northern Switzerland, found that just in 25% of them plants grow higher, and they run a bunch of statistical models to find that initial fungal pathogen abundance, a high initial fungal pathogen abundance, was a good predictor of this positive result. Then we also know, more or less from 15 years or more, that mutualism in mycorrhizas depends on the origin of the microbiome or on the phenotype of the plant. So the series of studies by Nancy and others show that there is sympatric adaptation, meaning that when the 3 components come from the same origin, the host, the mycorrhizal fungi, and the soil microbiome, plants grow more and they tolerate better some stressful conditions. So this is a very strong evidence of local adaptation between these 3 components. Finally, as we saw on Monday by Gu Feng's talk, there is the issue of the hyphosphere. So mutualism is dependent on third actors, so it's not just anymore, in my view, plant and mycorrhizas, but also the hyphosphere of the mycorrhizal fungi. This very nice study in new phytologist shows that there is a core microbiome across 3 very different ecosystems in China and also in a microcosm experiment where what is core is the gills, the functional gills of the bacteria, not so much the taxa. And this is very interesting for reasons that I will tell later on. This other study was a manipulative study. They separated these three components. They separated the host, they separated the mycorrhizal fungi, and the hyphosphere. Basically, what they conclude is that AMF fungi do not function in isolation of this plant and that the plant mycorrhizal symbiote recruits beneficial bacteria. So, with all this context dependency of the mutualism, meaning soil nutrients, meaning soil microbiome, meaning that is enhanced under sympatric conditions, and meaning that it depends on the hyphosphere microbiome, what we are suggesting is that the current way of thinking about cooperative interactions does not reflect this complexity. So what does reflect this complexity is a multilevel selection theory. So I'm going to talk about this very, very briefly. In short, the most agnostic definition of multilevel selection is natural selection occurring in 2 or more entities of the biological hierarchy. So from genes to organisms, groups of organisms, populations, communities. By the way, this is an article that I wrote for Frontiers in Young Minds, which was very difficult to write because the reviewers are 10-year-old kids, so it's very challenging. And with the drawing here, what we express is the basic concept of multilevel selection. At the individual level, selfish individuals beat cooperative individuals, but at the group level, cooperative groups beat selfish groups. This was established by Darwin himself in the Descent of Man. So there are three arguments in favor of this theory. First, that there is abundant empirical evidence. Second, the transitions in individuality. The third one, which is the focus of this talk, is that different entities across this biological hierarchy have different roles. I'm not going into transitions in individuality, but this basically means that a gene or an organism is already too complex. It's already too complex, so at least historically selection acted in things less complex than a gene or than an organism to reach such complexity. There is a lot of empirical evidence on multilevel selection, which is not so much discussed when you talk with evolutionary theories, but when you check the literature, you can see multilevel selection being used to explain the central dogma in molecular biology, also in cultural applications. As a matter of fact, the only woman that has ever won an economics prize was Elinor Ostrom, and she basically used multilevel selection framework. You know that both in economy and in ecology, the concept of tragedy of the commons is very common. But she showed that human populations don't reach the tragedy of the commons across the world, across many different cultures. And then there is the people in Georgia that do experimental evolution with genes, and this is some fantastic studies across thousands and thousands of generations of genes, and they basically reach a macroscopic multicellularity, which is amazing. So, since the last year, I am writing or analyzing this empirical data, and it's a lot. It's 375 studies that I have found. Some are experimental, some show direct proof, some suggest that the things that they are seeing could happen because of a multilevel selection process, And there are some 25 studies that actually measure in nature this process going on in many different kinds of organisms through a quantitative genetic method called contextual analysis. So, the third argument in favor of multi-level selection theory was that the expression units of selection has different meanings, which is what I will talk about. So this means that different biological entities have different roles, and we need to relearn what we understand as natural selection. Most people will think that natural selection, right, is just among genes, but when you read the 1970s literature of Richard Lewontin and Dawkins and so on, you see that there are different entities that have different roles. So genes are probably a replicator, which is the entity that gets differentially copied, but natural selection actually occurs in the interactors. So this is the entity that interacts through its traits with the environment, so replication is differential. And then there are some other phenomena, like these major transitions in individuality, which I mentioned earlier, which requires the reproducer, and the holobionts that requires the concept of the reconstitutor. I will not go very much deep into that, but this is to say that in addition to replication, there are other ways of inheritance that have been widely studied. The reproducer also includes concepts like epigenetics, for example. And then there is the manifesto of adaptation, which is the entity in which accumulated adaptations occur. And I will make this difference in a second. But in general, there is no reason to think that a single entity in the biological hierarchy needs to have these three roles. They are different functions. So what is this of the manifesto of adaptation? Elizabeth Joy back in the 80s made this differentiation between accumulated adaptations, these ones here, and then product of selection adaptations. So accumulated adaptations accumulate over time, the biology change, and this has been very well studied in Darwin Finch's beaks, while product of selection is a change in the phenotype through several generations, but the proportions, for example, of the white or black moths change, but not the biology. So I know that we have seen a lot of traits talks during the week, and this could complicate things even further, but I think that we really need to think about which mycorrhizal traits are accumulated and which are product of selection. Very quickly, I also want to mention that the classic genic view of evolution that most of people think about has very stringent assumptions, has very difficult assumptions. This was started by Fisher, and among the assumptions are that populations don't have epistases, that they don't have, that the interactions are totally random, that the environment is static, and this classical genic view of evolution is trying to explain how these accumulated adaptations could evolve. The other school that I think is more correct is the Wrightian school, where it just measures the things as they go on. Okay. And so to round the things up, when you make a search about natural selection and mycorrhizas, is very little what has been studied through many years. So here is a summary of those studies. Some people argue that pressures inside the roots and outside the roots are differential, which makes total sense. And then there are some studies showing endobacterial transmission through spores, for example, or showing nuclei level selection, which will be the next talk. But in general, it's a topic which has not been received too much attention. I can think of several of these entities which can be at least interactors in the definition that I just brought up, which could be the nuclei that are definitely interactors, and then the spore caps are also definitely interactors. So then the open questions. In general, I would like to know which entities across this hierarchy from genes and nuclei to holobionts are interactors, which entities are inherited across this hierarchy, which are the evolutionary implications of this core microbiome across the hyphosphere, again, which mycorrhizal traits are product of selection and which are accumulated, and then, importantly, what is the role of other microbiomes like root endophytes, phyllosphere, and other non-nutritional mycorrhizal functions. Overall, I think that we need consilience to answer these questions, to mix a bunch of different areas of knowledge, and we will schedule a seminar series in Twitter. Please pay attention about plant holobionts specifically. Again, molecular people, mathematical people, philosophy people, to have a better picture. My experience with philosophers is, and with biologists of course, is that we work in a bunch of systems that we don't define correctly, but of course the philosophers define a bunch of stuff without actually looking at it. So we need to mix these two things, and with that, thanks to the funders and thanks to you. Just 1 minute for questions, so yeah.
1 minute. We have a question. Yes.
Very nice talk, César. Thank you very much. I would like to know which is the role of invasive species here, because your main idea is this co-adaptation with the organism, the host, the mycorrhizal fungi, and the other rhizophilic organism co-evolving together, something like that. And we see that invasive species enter a new ecosystem and make new associations, and sometimes they are more benefit than natives. So what are your thinkings about this?
That's a good question. I think that because some of these, as Nancy will show us, some of these ideas suggest that adaptation could be very quickly, could be even in the lifespan of a plant, of the host. So I think that this could potentially explain the success of some of the invasive plants indeed. Yeah. And we need to go to the next. Thanks.
It's my pleasure to introduce Xander van Otterdijk.
Yes, this is my first time using a microphone. Can everybody hear me well? Yeah, it's working. Okay. So, first of all, I would like to thank Nancy and César for giving me the opportunity to talk to this beautiful workshop, and also kind of introducing me to this new concept. It's been really interesting to learn more about these concepts as well. So I will be expounding a little bit about what César has previously said, but I will be talking from a nuclear perspective instead. So, as we all know, AMF networks contain hundreds to thousands of nuclei in a single network. And these networks are completely aseptic. So they are all sharing one gigantic cytoplasm, basically. Additionally, these networks are able to fuse with other networks, and although we don't know what is the extent of nuclear mixing between different networks, this makes the concept of how selection occurs on the nuclear level, because you don't have this single nuclear life stage, or on the network level kind of blurry. So basically the concept of individuality becomes really blurry in AMF networks. So the spores, the asexual spores that they form, They are mainly populated by migration from nuclei all over the network towards these spores. And although there is some replication inside the spores as well, they're mainly populated by random migration of nuclei. And this suggests that spores actually function as genetic bottlenecks for sexual selection, basically. And because of this, we can actually view not just individual nuclei or nucleus as a unit of selection, but instead groups of nuclei can function as units of selection. The structures that AMF networks form are also highly complex. So we have all these different structures. This is a zoom out of a Dapistane network in a microfluidic chip. So we grew these networks under asymbiotic conditions. And you can see we have these really thick runner hyphae, and we have these really finely branched structures where nutrient absorption is facilitated, and then you also have the formation of secondary structures such as spores, for example, secondary spores. And in all these different structures, different requirements on gene expression are necessary, and it's also possible that you have different selective pressures on the nuclei in these different places as well. So what complicates this picture even further, so we already have these really complex networks basically, is that there is also constant flows in these networks. So my colleagues at Amsterdam in Amsterdam, they've looked a lot at, for example, lipid transfer through the networks and cytoplasmic flows. But what we should also consider is that there is also flow of genetic material through these networks, as you can see here. So groups of nuclei seem to migrate throughout the network in a regulated manner. And this all seems really chaotic, but still, AMF are a huge evolutionary success, so they're doing something right, basically. So if we zoom in a little bit more on these networks, we see that actually the distribution of nuclei throughout the network is actually really homogeneous. So this would suggest that actually the placement of nuclei is deterministic and not completely random at all. And we actually measured this. So we found, we used a tool called StarTaste, which is really good at segmenting star convex shapes, basically. So we could use it to quantify the number of nuclei in a network. And we found that there is roughly one nucleus every 5 micrometer of hyphal length. And although there are areas where the density is lower or higher, this is roughly the normal distribution. So now we have this complicated picture of complex nuclear dynamics, but still organization. But how has multi-level selection shaped the physiology of AMF and these complex nuclear dynamics? We could look at the genomes and the heterogeneity between nuclei to see at which level selection has occurred to shape basically their physiology. So we could look at the heterogeneity between different nuclei, for example, and because this influences the expected competition that we see between nuclei, and also the expected interorganismal conflict, so how much conflict do we expect between different nuclei in a network. And additionally, it could also be that different nuclei complement each other in function. This has been a really long-standing debate in the AMF field, but nowadays 2 different forms of nuclear heterogeneity are recognized. So the first form is nuclear heterogeneity to the constant accumulation of mutations in the network. And the second form is we have found a few dikaryotic strains that contain not one nucleotide, so not one distinct genome, but 2 different distinct genomes within the network, basically. And first I would like to talk a little bit about accumulation of mutation, because this can tell us a little bit about how at the individual nucleus level selection has shaped the genomes. And what we find is that the SNP density between these different nuclei is actually really low. So they have a really low SNP density in the coding sequence. And this was found by single nuclei sequencing of multiple nuclei and then extrapolating what if we had sequenced many more, what would be the SNP density, basically. but it's around 0.048 and 0.37 SNPs per kb in the coding sequence. And this suggests that there is some kind of purifying selection occurring in the network. Additionally, they looked at the ratio of synonymous mutations versus non-synonymous mutations, and they found actually that this is really low. So this is another sign of purifying selection occurring in the network, basically. But the mechanisms through which this occurs are really not clear. So it could be that there are elaborate DNA repair mechanisms, for example, or there could be targeted degradation of nuclei, or there could be also replicative rate differences between mutant and wild-type nuclei as well. So this suggests that at the single nuclei level, selection has shaped AMF, basically. There is also an example at a higher hierarchical level that selection has shaped it, and this is at the group nuclei level. And for this, we have to look at these dikaryotic strains where two different nucleotypes coexist in the same network. So this was discovered in 2016, and it was shown that these nucleotypes are quite similar, so they share 98% of base pair identity, but they differ in one loci, which is the mating loci. It's not been shown to be functional, but it's highly homologous to the mating loci in Bacillomycota. And they also have roughly around 10% of haplotype-specific genes, basically. And it has been modeled that if there was only this nuclear competition between individual nuclei, you would revert back from this dikaryotic state to again a homokaryotic state. But if there are selective pressures at higher hierarchical levels, so at the group of nuclei level or at the network level, you could stabilize this dikaryotic state, basically. So here we show some images of their artificial coloring, but of these situations where you have two different nucleotypes in one network, basically. But what are the emergent strains that selection could select for? So Vasilis has found in his previous lab in Canada some evidence that the nucleotypic ratio, so the abundance of these two nucleotypes, can change depending on the host that it's interacting with, and several abiotic factors as well. So they found that if the networks are grounded plant A, one of the nucleotypes is abundant, and if you grow with plant B, the other nucleotype is abundant, basically. They also seem to grow faster, longer, and further than the homokaryotic counterparts. So this suggests that there is that they are basically fitter and a little bit more selfish, as they are a little bit less mutualist as well, it seems like. I have to say that the comparison to homokaryotics is not completely fair because we don't have the parental homokaryotic strains to compare their fitness with directly, basically. But how could you transition from a homokaryotic stage to a dikaryotic stage? So in other organisms, such as Neurospora crassa, people have managed to do transformation protocols, and this allowed them to label the nuclei of these strains in two different colors, and then allowed them to cross, basically. And these fungi are also completely cyanocytic, so aseptate as well. And what they show is that by quick mixing of the cytoplasm, you transition from a homokaryotic stage to a dikaryotic stage. Don't know if it was really clear. If you see on the right, you see it becomes more green over time. Additionally, they showed that by looking at the heterogeneity in nucleotypes in spores and what was found in the network, that the heterogeneities were different. And this suggested that groups of spores or nuclei migrated to the spores. So again, enforcing this idea that groups of nuclei can function as units of selections instead. And now I want to look a little bit at my own work as well. So we've been trying to do something similar, but as you cannot really transform AMF networks, we've been trying to use another method, and this method is fluorescent in situ hybridization. And in this method, you use fluorescently labeled DNA oligomers to target either RNA or DNA in the cell, basically. And you can use it to visualize the presence of nucleotypes in the cell, And you can also show the nucleotide-specific gene expression, basically. And we hope this will help us better understand how nuclei function as reproducing units and the spatial regulation of gene expression, basically. So far we have managed to do RNA fish in these networks, basically. So here on the left, you can see an image in green where you have all these diffraction limited spots. So these are single RNA molecules. And in blue, you have the nuclei in the network, basically. And you can now link the density of RNAs to all kinds of spatial features, such as the distance to a nucleus, the distance to a branching point, and all this kind of stuff. And now we want to take it a step further and also link it to nucleotide-specific gene expression, for example. So we've been working for the last few months to make DNA fish probe libraries that are specific to either of these nucleotypes. We've just finished them, now we're ordering them, hopefully it will work. So that's the next step. So with that, I want to finish my talk, but I want to leave the audience with some outstanding questions as well. So first thing I could think of is, has this ability to regulate nucleotypic ratios, has this alleviated regulatory burden on the gene level? So is there less regulation necessary at the gene level because it's now regulated at a higher level, for example? The other one is, Bacillus has tested hundreds of strains, and most of them seem to be homokaryotic. So what is so especially hard to transition from a homokaryotic stage to a dikaryotic stage? And why is two nucleotypes in one network, why is that the seeming limit? Can there also be presence of trikaryosis or more? Or what is so difficult about this? And lastly, how can the network topology, so the number of branches, for example, actually influence the maintenance of diversity in the network, basically? And with that, I want to thank my wonderful lab, firstly, for helping me prepare these presentations, and you also as an audience for listening.
So we have 4 minutes for questions. Ian?
Thank you for this very nice talk. Actually, we don't know that dicaryons are rare. Yeah. What we know is that we don't have many in culture. It doesn't mean they're not rare in nature, right?
No,
I think actually using the probes you mentioned, I don't know if it's possible to get spores out of the soil and look at the nuclei and then try and get an estimate, because it may be just some bottleneck for getting them into culture, right? And it may just be that they don't grow well in culture. We actually made what we think are dicaryons in our lab, many, many years ago. If you look at the paper in the Phytologist, 2008 or 9, where we actually put AMF together and made crosses, we had evidence of cultures that had alleles from the two parents. So we sort of made them in the lab, but we couldn't maintain them any longer. Okay. So perhaps they occur in nature, and perhaps there's lots of swapping of nuclei, but the bottleneck is the culturing. So just an idea.
Yeah, and about the fish probes, you really need high resolution genome sequencing to make these fish probes work. So you really have to make them specific for every strain that you have. So it's not as easy as just getting a sample and testing it with your fish probes, basically.
But you don't need to picture nuclei to quantify the network.
Yeah. Good, that's also a possibility, yeah.
Someone else has a question? Camille?
So I was wondering if you have information or suggestion about the fact that maybe some species would be more likely to have this dikaryotic stage than others. For example, in Basidiomyces, it's well known that some species are heterotypic and some species are monotypic. So do you think that's also some space happening?
I don't know. I think one crucial thing is the ability to really quickly mix, have this cytoplasmic mixing. That could be a really important quality to go from homologous to dicarial. I also mentioned that you can have some genomic diversity, but it cannot be too much because then you get these genomic conflicts within the organism. Otherwise, it could be that the genomes really nicely complement each other, for example, which leads to dikaryosis. So I think there are multiple ways to go from monokaryosis to dikaryosis, multiple factors to consider.
Yes, go ahead, please.
Do you think there's any evidence that the translation machinery, so the ribosomes and the various mRNAs, move around with a parental nuclei, or are they kind of moving independently of nuclei, and how would that affect selection?
Yeah, we have, so it's very preliminary, but we see at least that the density around nuclei of RNAs can really differ, basically, and there are patches where the density of RNAs is really high around the nucleus, and it's very more sparse, so it leads to heterogeneity in gene expression between different nuclei. But whether they travel together is hard to tell because this is really a fixed cell method, so you're always frozen in time, basically. So I think you would have to couple it at least with the flow dynamics before fixing the network and then doing fish to see whether things travel together. I think, but I don't know if that
Okay, thanks. Next we have Letian Wang from China University.
Okay, thanks a lot, Professor Nancy and César, for giving me this opportunity. And my name is Letian Wang, and I'm from China Agricultural University. And I'm very pleased to introduce our work about the hyphosphere of hyphosphere co-microbiome of fungi and their interaction, their function in organic phosphorus mineralization. And here's the outline. Please.
Yeah, it's an extra.
Okay. Fungi and bacteria live together in many environments, and their interactions are key drivers of many ecosystem functions. And studying these interactions is a hot topic in microbial ecology research. And as we know, fungi are widely distributed in terrestrial ecosystems, and it provides many benefits to host plants. And the actual radical hyphae of fungi host numerous bacteria, and known as the hyphosphere microbiome. These bacteria feed on hyperoxidates and interact closely with fungi. This just provides an ideal system for studying fungal bacterial interactions in soil. And advances in sequencing technology have given us a deep understanding of the hyphosphere microbiome. Its composition is different from the bog soil and the rhizosphere, and influenced by biotic factors like host plant, fungal species, or abiotic factors like soil type, soil nutrient levels. And a lot of studies found that the microbiome linked to animals or plants have both variable and shared components. The shared components is the core microbiome, which I believe to be the most important functional microbial partner for that host. And recently, studies have found that the hyphosphere of fungi have a core microbiome, and this core group is independent of soil conditions and fungal species. But our understanding of the hyphosphere core microbiome is very limited. But it shows great promise for understanding crucial fungal bacterial interactions in the soil. And recently, most studies focused on one fungal species with one bacterial species or bacterial community. However, in the field, the situation is much more complex. Different fungal species colonize the plant roots at the same time, and their actual radical hyphae recruit diverse bacteria, creating a complex interaction network. So there is raised the question, how well do the existing studies reflect the real patterns of fungal bacteria in the field? Therefore, we put forward two questions. First, in the real world, does the hyphosphere of air fungi have a core microbiome? And second, if yes, what's the function of the hyphosphere core microbiome? We mainly focused on the organic phosphorus mineralization because air fungi cannot do this on their own. And following these two questions, we face some problem. First, as we know, air fungal hyphae is very slender. So how to collect air fungal hyphae directly from the soil is very difficult. And the second problem is that how to identify the core microbiome and its function accurately. We know due to the the high diversity of bacteria makes it difficult to single out the core group from the rest, and hydrogen further link them to the functions. So to solve the problem of collecting hyphae, we created a method. We developed a method to induce air fungal hyphae in the field and isolate and collect them in the lab. Firstly, we created two types of ingrowth tubes. One allows air fungal hyphae to mate, and the others keep them out. And in the tubes, we placed some membranes to collect the hyphae. The idea is that when air fungal hyphae enter the tubes, they will accumulate nearby the membrane, making them easy to collect. And we buried these tubes in the field and left them for a period of time. We retrieved them for collecting hyphae and test their purity. And using this method, we conducted a field experiment. And we chose 3 long-term experimental sites toward humid, semi-arid, and arid conditions, different soil types, and 2 plant species. Firstly, we identified the hyphoelasticity in the tubes. We found no hyphae detected in the tubes where air fungal hyphae were excluded. And in air fungal hyphae permitted tubes, we saw a lot of hypho growth. This indicates that our 2 types of ingrowth tubes efficiently control hypho growth and created soil environments with and without air fungi. And then we calculated the difference between 2 types ingrowth tubes with and without air fungi. This helped us see the effect of air fungi hyphosphere bacterial interactions or organic phosphorus use. And at each site, when air fungal hyphae into the tubes, the soil organic phosphorus content decreased, and phosphorus activities increased significantly. This confirmed that air fungal hyphosphere bacterial interactions prove the soil organic phosphorus mineralization. And to identify the hyphosphere core microbiome, we classified bacteria according to their presence and relative abundance. First, we identified site-sensitive OTUs. They are OTUs whose abundance varied across experimental sites significantly. This indicates the impact of site-specific conditions on the bacterial community. And then we identify the hyphosphere specific OTOs. These OTOs have a higher relative abundance in the hyphosphere than the bulk soil across all sites. And this can help me identify help us identify the bacterial members which have a robust and strong connection with AM fungi. And finally, we identified 82 HSOTOs. And these HSOTOs have several characteristics. First, their relative abundance is quite substantial, over 1/5. And the second, most HSOTOs are stable, do not belong to SSOTOs. And at the class level, most HSOTOs are conserved, mainly including 3 classes. And we also found that most HSOTOs have a positive correlation with phosphates activities. And based on these informations, we believed that these HSOTOs may be the core of the hyphosphere microbiome. And we further constructed the fungal bacterial interactions network, and we mapped HSOTOs and SSOTOs into it. The star indicate HSOTOs and the green colors indicate SSOTOs. We found that HSOTOs formed a distinct, stable module, which is distinct from SSOTOs. And we also found that the relative abundance of this module is positively related to the phosphates activities. And this reinforced our idea that these SOTOs could be the hyphosphere core microbiome. And to validate the result from field experiment, and we conducted a pod experiment. We mainly focused on 2 factors, with or without AM fungi, and four different soil types. And we used the completely same method to analyze the pod experiment. And we found that for both experiments at the class level, HSOTO were dominant by the same 3 classes, mainly including alpha proteobacteria, actinobacteria, and gum proteobacteria. And in the pod experiment, we found that the network distribution pattern of the HSOTOs was consistent with the field trio. A stable, unique, and organic phosphorus related module was also detected. And based on the existing studies, we further explore the potential function of the hyphosphere core microbiome beyond organic phosphorus mineralization. First, we speculate that the hyphosphere core microbiome may assist AM fungi acquiring organic nitrogen. And some bacterial predictors may act as the immune system of extensive hypho networks. And some core members may promote the hypho growth and the formation of the spores. We are providing fatty acids. Of course, these ideas need to be explored in the future. And to conclude, our work highlights 2 findings. First, different AM fungal species share the core hyphosphere microbiome, and this core group is conserved at the class level. And the hyphosphere core microbiome compensates for the missing function of AM fungi with respect to the organic phosphorus mineralization. And for future perspectives, we mainly focused on 2 questions. First, as Professor Nancy and César provided, when talking about AM symbiosis, how do we define the evolutionary unit? our result indicated that besides plant and air fungi, hyphosphere bacteria should also be included. And the next second question is, what mechanisms underlie the formation of the hyphosphere microbiome? And we try to understand this question based on the fate of phosphorus. During pathogenesis, total phosphorus content decreased and the organic phosphorus content increased. But air fungi cannot use organic phosphorus alone. As we know, phosphorus is a currency which air fungi use to exchange for carbon with plants. So therefore, the formation of the hyphosphere microbiome with phosphorus stabilizing ability is crucial for the stability of AM symbiosis. And many thanks, thanks to many people. And this picture is Mycorrhiza group in China Agricultural University, and this is my advisor, Professor Gu Feng. And thanks a lot, for your attention.
Many thanks. So we have 3 minutes for questions. I see a hand here.
Oh, perfect. Yeah, that was a really cool talk, and I love that you figured out a device to sample the hyphosphere microbiome so effectively. I guess my question was inspired by your use of the framework of core microbiomes and also of interaction networks, which connote these sorts of biotic, positive feedback loops between these biotic interactions between bacteria or other organisms. I guess I just think about so you've identified a core microbiome across all of these field-based samples, but do you have any way of verifying experimentally, maybe with growing AMF in sterile conditions and then adding particular bacterial strains to understand if those bacterial strains actually do facilitate each other's persistence in the hyphosphere, if there are actually these biotic interactions that are implied by the core microbiomes framework, are they actually happening?
I don't sure if I catch your question. You mean the function of hyphosphere microbiome?
I guess what I mean is, do you have any experiments planned or any data to suggest that there are actually biotic interactions happening between the members of the hyphosphere microbiome?
Yeah, the interaction between air fungi and the hyphosphere microbiome.
Or just members of the core microbiome, like bacterial members?
Yeah, this is a good question. And we have isolated some core members, but the work about interactions, we don't do this, but this is a future work we need to work on. Yeah, thank you.
But there is correlative evidence. There's good correlative evidence from your phosphorus concentration in those cores was highly correlated with the phosphatase enzymes, which was also highly correlated with those core bacteria. So I think there's solid correlative evidence.
But there is also experimental evidence in that paper you commented. Maybe there is one more question. Yeah, please.
Thank you. Yeah, really nice talk. Got a question about, we extract the core microbiome from the interrelated mycelium system. I think the mycelium must be a mixture of live and dead. And I guess the microbiome on the live sphere versus the dead sphere should be different.
Yeah.
And the storage should be much different as well. Have you always considered in the future to separate them, or how that can influence your sort of study here, framework here?
It's a very important question, and I agree with you. The death and the life have their hyphosphere microbiome may be different. Yeah, but as we know, I don't know how to differentiate them. Maybe in the future we can accomplish it.
Okay, I guess, thank you, we're out of time now. Thank you so much. It is my great pleasure to introduce Jim Bever, who is a very long-term friend of mine. I was just thinking, we met at a conference when we were still graduate students. And now look at us. Just thank you so much for participating in this meeting.
Yeah, thank you for organizing the session. I really have been looking forward to it. So I'm very happy to have the opportunity to describe my view of multi-level selection in AMF. I'll wait a minute before I get to it. Cool. So we know that multi-level selection is important because of the conflict between levels. So we can see evidence of it when we have direction of selection at one level that conflicts with the direction of selection at a second level. So, for example, when with replication of genes within nuclei versus organismal phenotype, we know that there are conflicts between those levels of selection because there are transposons that spread, have differential advantages in replication that do not confer benefits to the host. In that conflict between selection against transposons at the organismal level, selection for transposon at the replication level determines the overall abundance of transposons. And so within AMF, we just talked about how there could be different levels of selection for nuclei. So you have selection for nuclear replication, you have selection for function of the hyphae, and so you have the possibility that there could be selfish DNA that, or selfish nuclei that spread via replication advantage but don't confer a functional advantage or are selected against at the hyphal function scale. So similarly, you have possibility that that some mycorrhizal fungal types, genotype species, have competitive advantage at a local root scale, but they have a disadvantage at the larger group scale, which this could be related to, oh, this is really going down faster there. There's supposed to be fungi. So cheating fungi, for example, could have a non-beneficial fungi to the plant could have a competitive advantage at the local scale, and then plant dynamics, which would determine relative abundance or relative success at crossroot scale, could determine the stability of mutualism. And I'll talk about that. And then you have these associations at a larger scale between plants and fungi, which could be advantageous and could be a unit of selection, which we've been talking about as a holobiont. In that sense, we would know it might exist if there are differential advantages to the mycorrhizal fungi that accumulate with one host relative to that of a second host. So within AM fungi, we do know that there's a lot of selfish DNA in more than most organisms in the genomes that have been replicated, so that appears to be important there. We don't know a lot about the differences in levels of selection between nuclei in the organ and hyphae. We have had some really cool possibilities earlier, and I'm going to talk about the last two possibilities that I've worked on in my work, and I'll just note that these levels of selection are not necessarily independent, and so in particular, I'm going to talk about a condition for the holobiont. Well, this is way off. I apologize to everybody, but so I'm just going to point. So I'm going to talk about a condition for the holobiont possibility, and in the course of that, introduce evidence for cost of mutualism in cheating fungi, talk about what stabilizes that, and then use the dynamics of that system to gain inference on the likelihood of the holobiont of the plant-fungal combination being a coherent unit. Okay, so when will plants and fungi act like a holobiont? So if you see associations between plants and fungi, where the bar is the strength of correlation, this could be an interaction network. This could result from symmetric fitness relations, which is the assumption that we would need for the plant-fungal combination to act like a unit of selection, where they have shared interests. But it's also possible that we have, so this symmetric fitness relations would generate positive feedback, and in this context, the holobiont may be an appropriate perspective on the dynamics. It's also possible that we have highly asymmetric fitness relations, that the correlation is due to the accumulation of the fungi, which then don't actually deliver the greatest benefit to the hosts in which they have the highest fitness on. This would generate negative feedback dynamics, and in this case, the holobiont would not make sense because we get continual reshuffling of the fungi in the roots of the plant. So then, when do we get positive versus negative feedback within the mycorrhizal mutualism? So I didn't say, in this example here, the negative feedback, that's actually the dynamic that happens. I measure those fitness relationships. There's highly asymmetric to generate this negative feedback. And we can use that to gain inference on the conditions for positive versus negative feedback, because we get this negative feedback because of a negative correlation between the effect of the fungi on the fitness of the plant. This is this axis here, and the relative fitness of the fungi on that plant. So the fungi that best promote the growth of Plantago have low relative growth rates on Plantago. So that negative correlation is what we expect from their cost of mutualism. And what we can infer then is that when the cost of mutualism drives fungal dynamics, then we will likely get negative feedback. So the cost of mutualism is what we expect from armchair theorizing about AM fungi. It's going to be energetically expensive to the fungus to acquire phosphorus, move it down the hyphae, and deliver it to the host. So the fungi that deliver phosphorus at a high rate are incurring greater cost, and therefore they'll be able to apply less of the plant's carbon to their own fitness. So they would have lower fitness relative to fungi that deliver phosphorus at a lower rate. As a result, they are selected against, so that over time, non-beneficial fungi accumulate and benefit to a given host declines, and that's what we observe in this example of negative feedback. So what prevents this from degrading the mutualism? So this is the dynamic that happens within a root, within a local scale, when there's a cost of mutualism. So the question is, can the between-root dynamics overcome this cost? And that'll be the case when the most beneficial fungus is assembled in a patch that has the most beneficial fungus, receives the greatest benefit from the host, where the plant is preferentially allocating resources to that host, and we can test that with split root systems. So we have plants and fungi from the same system. In this case, Chlordiolormus candida is a much better mutualist than Gygospora decipiens. These are all plants and fungi that co-occur together. So we split the root, we label the carbon, and we see after 4 days, 4 days following the labeling, we have significant preferential allocation of that carbon in the roots towards the beneficial fungus. So we can look at the context dependence of this preferential allocation in this system. So in this system, we have the ambient phosphorus, and we increase it up to 4 times phosphorus. And here, the benefit from the beneficial fungus declines from the plant being 6 times bigger to being roughly 2 times bigger. The non-beneficial fungus never improves the size of the plant. It's never beneficial, it's never detrimental. So if we look at preferential allocation, it declines with increasing levels of phosphorus. So in this experiment, Baoming Ji was successfully labeled phosphorus in the two sides of the pots, P32, P33. And so we can look at phosphorus uptake in this system. And we're looking at the differential uptake from the beneficial fungus relative to the beneficial fungus. So the beneficial fungus is providing 16 times more phosphorus in low phosphorus condition, but the advantage of the beneficial fungus declines as we fertilize these systems. So in this system, we measure the carbon going down, we measure the phosphorus coming up for every plant, for each plant fungal combination. And we can say that the actual phosphorus exchange rate between the plant and fungus doesn't change as we increase the fertilizer, as we increase fertilization. So the carbon to phosphorus exchange is constant, the fungal exchange rate is constant. What's changing is the volume of carbon invested in the different fungi. So the plant has the plastic behavior, the fungus was not plastic in its behavior. So does this preferential allocation result in increased fungal population growth rate? So we return to this split root system, and now we have the two beneficial fungi together, two non-beneficial fungi together, and we have the beneficial and non-beneficial fungi on different sides of the roots, separated, and here we have the beneficial and non-beneficial fungi mixed into each other. So high spatial structure, no spatial structure. And the question is, does the spatial structure alter the fitness of the fungus? And this is the fitness of the two fungi as measured by total spore volume per unit soil volume. And so the beneficial fungus has high fitness provided it's not mixed in with the non-beneficial fungus. The non-beneficial fungus has low fitness except when it's mixed in with the beneficial fungus. So this result here, where the beneficial fungus has higher fitness than the non-beneficial fungus, this is what we expect from preferential allocation. So this result is what we expect from the cost of mutualism. In mixture, the non-beneficial fungus has a competitive advantage. So this is what we expect from there being different conflicting selective dynamics within root in the between root scale. So the between patch dynamics can overcome the cost of mutualism, and we can infer that spatial structure within a plant's root system is essential to the maintenance of the mycorrhizal mutualism. So the dynamic is like this. So if we have no spatial aggregation of the two types within a root system, the non-beneficial fungus will increase everywhere, and the beneficial fungus will ultimately be lost. But if we have patches of high abundance of the beneficial fungus, then the plant will invest more in this patch. Mixed patches, the beneficial fungus will decline over time, but this is going to become denser, and so when there are new root systems, they're likely to be colonized by the beneficial patch because of the higher density relative to the non-beneficial patch. So here, I'll just note that we're assuming relatively limited dispersal at the scale of a plant root system. So if we look at the test here, we didn't allow dispersal between the two sides. We varied initial spatial structure, we didn't vary dispersal. So we can vary dispersal in a pot by having pots without barriers, pots with barriers that have holes, and pots that have barriers without holes. Dispersal is higher here, lower here. And then we can vary the initial spatial structure within the pot. So we have mixed pots and pots with higher, oh, the color's terrible. It's supposed to be blue and red, anyway. So, sorry about that. So, and where the colors are, the fungi are separated or they're well mixed. So these are those pots. And here we're looking at the fitness of the beneficial fungus when it's separate and when it's well mixed. So they have higher fitness when they're initially separated from the non-beneficial fungus, and when there are barriers to their dispersal. They have the lowest fitness when they're stuck in a tube with the non-beneficial fungus. So the non-beneficial fungus has high fitness as long as it's initially well mixed with the beneficial fungus, and then it increases without limits to dispersal. So this then affirms that we have this difference in multi-level selection, difference in compatibility that depends on the spatial context and is consistent with the conflict between the competition among plants, among fungi within a root and the dynamics of between roots. It also gives a way to think about and expectations for the dynamics of these multi-species communities and the likelihood of positive feedback. So we're likely to have negative feedback, for example, when the cost of mutualism dominates the fungal dynamic, and that's going to be the case when we have low spatial structure within the root system, when we have high soil resources, and when we have this significant cost of preferential allocation to the plant. And this is something that comes out of a model with Brian Senator and I. We're likely to have positive feedback, and in this case, the holobionts is unlikely, we wouldn't expect the holobiont to be a unit of selection. We're likely to have positive feedback when preferential allocation dominates the mycorrhizal fungal dynamic, which will be the case when we have high spatial structure within a plant root system, and when we have high above ground resources relative to below ground resources. We saw revaried light, for example. And when we have a correlation between the ability of a plant to preferentially allocate and the responsiveness of that plant. And in this case, the holobiont is a possible unit of selection. So I probably talked too fast. What time?
3 more minutes.
Well, the benefit is that on one hand I'm done, and I do want to advertise the Invent poster, which it's downstairs, but I think with Tara as the associated with that. I did a terrible job with this picture. It doesn't include the people who actually did most of the work I just showed you. And I have it because Rob did this work that I didn't think I had time to tell you about. I also did want to tell you about this micronet thing. My slides suck. So I did a big feedback experiment. Rob did a big feedback experiment. So we measured the amplicon speed. So we have lots of fungi from the lab, from mixed prairies, 39 different plants that vary in plant life history, and plant phylogeny and life history. We did the feedback test. So this is a ton of work. Rob Ramos did an awesome job with it. We measured, so this is the phylogenetic structure of the AMF community. So we have significant variation due to plant phylogeny and significant variation due to plant life history. So this notion of phylosymbiosis is correlated, is parallel to the notion of holosymbiont in that this is inferred to be evolutionary advantageous when you see it. So we see phylosymbiosis at the scale of the abundance of these fungi, of fungal species. We also see at the scale of genotypes within those species. So here, these are the relative abundance of ASVs that belong to these species, and they are significantly varying with phylogeny and with life history. So does that result in positive feedback for these prairie species? And when we look across phylogeny, here is a strength of feedback as a function of phylogenetic distance, and it's basically flat. This is the best line. It's not significantly different from 0. When we look across the plant life history, we see, on average, positive feedback between pairs of early successional species or between early and late successional species. So this would be the dynamic relevant for succession, but not between late successional species. So late successional species are very responsive to mycorrhizal fungi, but they're all good hosts to mycorrhizal fungi, so they actually do equally well with each other's or their own fungal community. So I'll stop.
Yeah. Sorry, we don't have time for questions, but if you stick at the end, we can stay some more minutes if you want to ask something to Jim. So now it's my great pleasure to introduce to you Nancy Collins Johson from North Arizona University. Just a little anecdote and a little commentary. We started talking about this in ICOM 10 in Merida, and it's very nice to see that it has become this, and we have projects and publications going on. So please go to ICOM because it gives you a lot of opportunities.
It's my great pleasure, and I want to thank César because, you know, when you get to be my age, you get kind of tired, and sometimes I just don't have the motivation to do what needs to be done to, you know, finish papers and things like that. But he has energy, and we have questions, and with his enthusiasm, I'm really motivated to continue to work and move forward. It's been delightful. So I am going to talk about, I'm going to try not to just wave my arms, but again, this is more of a whole view of evolution and just some ideas. So again, I want to talk about something that César and I are working on. It's this concept of functional team evolution, and it builds on some of these discoveries of all the complexity in the microbiomes and in the root and around the roots and the rhizospheres. So we have some wonderful quotes from Charles Darwin, and many of us know this particular passage where he refers to an entangled bank, and I just love this part. So different from each other and dependent on each other in so complex a manner have all been produced by laws acting around us. And so he recognized this tangled bank, and here's an artistic rendition of the tangled bank. And that's a microscopic, we have a microscopic version of that we study. So today I'll just briefly talk about the evolution of the theory of evolution through natural selection. I'll talk about this idea of plant adaptation through functional team selection, and then I'll provide some empirical evidence that this might be happening. So here we are in Great Britain, and it's, I think, appropriate for us to reminisce how these two fellows were discovering some really interesting insights. All these ideas came together, and through their really naturalist work and observations all over the world, they came up with a theory that has just been really driving our science ever since, the evolution through natural selection. And the gist of it is that individuals within populations vary, the variance is heritable, and the variants that best survive and will replicate are passed on to future generations. So this is really the core paradigm of evolution through natural selection. And of course, after they did their work, more researchers have discovered the genetic mechanism of heredity, and this has been articulated into the modern synthesis, where genetics was incorporated into this very important framework. And this synthesis continues today. There's the extended synthesis that is an ongoing process. And I think that we can add some more insights to what's going on and recognize that we have to do a little bit more synthesis to account for some of these observations that weren't visible, not even very long ago. We had no idea that plants and animals were really not individuals at all, but they're communities. Like, so what's that all about? We got all these communities, and we've been calling them individuals all this time, but we have to recognize that they're really holobionts. We also know that epigenetics is a really important phenomenon. So it kind of takes the only genetic mechanisms for heredity down a notch, because it's not always just genetic. We also know that genes are very promiscuous, and they'll move from one unrelated organism to another organism. this horizontal gene transfer really kind of messes things up. And we've got to accommodate that. And finally, we have very good evidence that microbiomes can generate adaptations in both plants and in animals. So if I was to just slightly wordsmith what they did, I would say that holobionts within a population vary, and the variation is heritable. Now, they didn't know it was just genetic, they just knew it was heritable. But the first synthesis really focused on only genetic mechanisms. But now we know that there's non-genetic mechanisms as well. And the premise that the variants that best survive are passed on to future generations, that's still rock solid. So I want to talk now about this idea that César and I have been working on. It's this idea of functional team selection. And it's just a framework that we think might be helpful. And again, if plants are really communities, they are kind of like the roots of a plant is its own entangled bank. And there's many different organisms all interacting, and they all have their own life histories. They're all evolving, and we know that variation within each of these organisms can be inherited genetically, but especially in a viscous material like soil. There's not we just heard about Jim Bever, there's not a lot of mixing, and from one generation to the next you have a buildup of certain populations of microorganisms and AM fungi. They can build up, so there's huge legacies. That's inheritance. It's not genetic inheritance, but it's inheritance, and it's something we should not ignore. So we know that these huge scaffoldings of hyphae are present in the soil, and these scaffoldings create places where bacteria and other microorganisms can hang out and exchange DNA with each other and interact with each other. So they're functional teams that are developing over time spatially. And these groups can work together to increase the primary production of the host plant, which is it's pumping substrate below ground to all of these organisms. So it's just really a very important selection tool. Now, both Darwin and Wallace were pretty much into competition, but I would argue that actually mutualism, working together, is as strong or perhaps a stronger force creating evolutionary change. And I don't know if it has something to do with the Industrial Revolution, of course, that was going on in Manchester at the time, but there was a lot of tooth and claw struggle for survival going on in the economics at the time. But I think now we recognize that actually teams that work together can often beat teams that are, struggling and competing, turns out. So we know that it could be a selection for mutualism. So there could be functional teams of unrelated organisms that are hanging out in the same place at the same time. And we also have seen in the talks this week that plants can actively curate the composition of their microbial communities. They can recognize who's there, and they can send signals. They can do devious things like have a root that just tests the waters, and then if it doesn't look good, then stop. So they have ways. We don't know what all the ways are, but they have ways. They can curate their microbiome. They can curate their mycorrhizal fungi. They know who the good guys are. And now we know that the mycorrhizal fungal hyphae also curate their bacterial friends. So there's a lot of recognition going on, and we're just now having the tools where we can start to recognize that. The fungi can also curate their hyphae. And this ability is heritable, both genetically and, as I mentioned before, non-genetically, because these populations of the ones that are being selected for are going to be increasing in the rhizosphere. So that would be a legacy effect. So here's a diagram that sort of lays out functional team selection, and I want to thank Cesar's friend for this illustration. So here are some plants growing in some soil at some time in the past, and we're illustrating beneficial winning AM fungi in this gold, and then the resident AM fungi in this blue color, and they're just in the soil. Notice these plants are not doing particularly well. This was sometime in the past. We also have other microbes. This can be bacteria, archaea, nematodes, viruses, you name it. There's resident ones, and then the winning ones are this bright pink color. All right, so these plants are growing. They're not doing so well, but they can select the good ones. And over time, these good ones could be building up in their rhizosphere. It's a viscous substance. Their populations might freeze in the winter and come back in the spring, but they're building up over time. Notice the plants are starting to do better. So here's a strong selection for mutualism. The good guys are getting more carbon. It's a mutualistic selection, not just competition. And in the present, these are clearly enriched. And so this is essentially the mechanism. It's being driven by a host plant that is supplying rewards to organisms that are helping it. And I think we've talked a lot over time, over the years, that the function is context dependent. And so microbes can generate, we have seen it, we've all seen it. We would like to talk about the beneficial ones, but it turns out there can be neutral or even detrimental microbiomes, and we can't ignore them. Why is that happening? Well, it can be caused because the environmental conditions influence the fitness outcomes. So here's that same diagram. Here's the time in the past to some time in the present. Here is limited soil resources, and the same schematic if the resources are not limited. There's no selection for mutualism. There's no allocation to the good guys. And I was so happy to hear about Jim's work showing that high soil resources, they don't see the selection. So this follows that. So plants can select for functional teams that ameliorate resource limitation or stress, and could be even pathogens. But if that stress is removed, then that selection is gone. And so the removal of these limitations can shut down that selection force. So I want to give some empirical evidence now that this could be happening. These are two publications from a PhD student that worked with Matt Balker, and Matt Balker was my first PhD student, and so I guess that makes me Michael Remke's grandmother. I kind of like that. So these studies, I think, are some good empirical evidence. They involve a grass that's very abundant in our part of the world. It's called Bouteloua gracilis. It's a C4 grass, and it does very well in the arid southwest United States. And what Mike Remke did was he collected Bouteloua seeds from two different locations. One location was wet, and the other was quite a bit drier, and they weren't very far apart. It was an elevation thing. So they weren't far apart, but they had very, very different environments, like double the precipitation. And then he grew these grasses, the different genotypes or ecotypes, with their home or sympatric soil organisms, or the opposite soil organisms, so either a sympatric or allopatric way. And then he killed the plants slowly by not watering them. Either slowly or kind of really fast. And then he monitored how quickly they died in their biomass at the end. And what he was able to show, this is a soil biota effect, it's essentially just a response ratio of a plant with the inoculum from either the wet site or the dry site, inoculated with its own sympatric or allopatric microorganisms. And they either had extreme drying or moderate drying. If it's above 0, it was a positive effect, mutualism. If it was below 0, it was a growth depression. And if it's brown, this inoculum came from the dry site, so the dry plants inoculated with their own dry site microorganisms were much benefited. If it was an allopatric, they had growth depressions. This was very strong when we really dried it out. It was cruel, it really was. And then this was a slower drying, but it still had a positive effect. Here's the wet site population. Its sympatric population of inoculation also had a positive effect. The allopatric caused quite a growth depression. Home is best. And if we looked inside, of course, this is whole soil inoculum, so it includes bacteria, it includes everything. If we want to figure out what the mycorrhizal fungi are doing, the root colonization, you can see there's a positive relationship with arbuscules, which are the exchange organs. They're very highly correlated with good things. I would call vesicles kind of a selfish organ, and it's all about the fungus. And they were negatively correlated, so that fits too. So then that's fine and good. It was done in the greenhouse, but to really understand what's going on, you need to go in the field. Mike Remke is a young man and very ambitious, and so he grew not just, he got 2 different genotypes of this grass, and then he grew them at their local sites with the local sympatric inoculum or allopatric inoculum from 6 different sites across an elevation gradient. And these elevation gradients have very different climates, a huge change in temperature. And he filled pots with the local soil that was sterilized, then he added back plugs of live soil, and then he had the different genotypes. This is a cartoon of the experimental design, so we had all different combinations of allopatric and sympatric inoculates. Whole soil inoculum. As you go lower, it gets really hot, 3 degrees hotter. Go higher, it's less stressful, 3 degrees cooler. And we were so thankful that Martina Janskova was a visiting researcher in our lab at the time, because she brought some of these samples home, and she was able to confirm that these were in the field for 3 years, and after 3 years, the inoculum was still in those roots. I think it's because they were inside the pot, but we just really needed to know that. So it probably wouldn't have lasted if it wasn't in the pot, but that was important to know. And so here's the results. Here's the source of the seed was here. This is the standard that we were, the home all sympatric genotype living in its own location with its own inoculum. And essentially what we can see is if the plants were inoculated with their sympatric inoculum and they were brought to a more stressful environment, they actually could grow as well as if they weren't brought to a hotter environment. But as we predicted, if it became less stressful, it didn't matter what the inoculum was. It was irrelevant, the source. So the inoculum source was irrelevant if the plants were moved to a less stressful environment, but if they were moved to a hotter and drier environment, then the sympatric microbiome generally grew as well as the reference site. So this suggests then that sympatric organisms can ameliorate the stress caused by climate change. And again, I just want to reiterate that there's a lot of exciting work coming out that shows that streptomycetes and other bacteria are very involved in this interaction in the hyphosphere. And here's some work coming from Australia that these could be very important in drought stress. So I really think what we should be doing is not just looking at AM fungi, but also the bacteria. So just to wrap up, the evolution through multi-level selection involves holobionts, and that these could be functional teams in the tangled bank. Functional team selection provides a useful framework for understanding the processes involved in creating and maintaining beneficial microbiomes, and that can be in agriculture or in ecosystem restoration. A lot more research is needed to understand all the mechanisms. I was going to take the word physiological out, but all mechanisms by which plants and mycorrhizal fungi curate their microbiome. And I really think, I didn't talk about this much, but I think we need to try more and more to look at the whole system and go in the field. So I'll end here. Do we have time for any questions?
A couple of questions.
Couple of questions.
Yeah, we can have that. Yeah, over there, please. There's two guys.
Thank you for that very interesting talk. Just maybe a remark for discussion that occurred to me. When we talk about evolution and kind of fitness to the plants, in your data you look at biomass, right? It's kind of assuming that biomass will automatically translate into the next progeny, but maybe to really think about Darwinian fitness of the plant, we should look at the seed count and seed biomass, seed viability as a more sort of close measure of the plant performance, and really thinking about selection in that way.
Yeah, and actually we did have seed biomass in that paper too. But these plants, I think, are very old and clonal, and so their vegetative biomass is a pretty good measure too of their longevity and, you know, how much they can spread vegetatively too.
Okay, the last one.
Thank you very much for this very nice talk. I would like to ask you your opinion about using bioinocular in the field in agriculture, since you seem to show that the context is very important, but many companies are selling microbiome, mycorrhiza or some strains, but is it possible to make some solutions that are diffusible in different environments, or do you think it's going to be a failure?
I don't want to be pessimistic, but I think it's going to be a failure. Thank you for that question.
Okay, thank you very much.